Okay, hello guys. My name is Lydia Okyere and I'm doing this presentation, a work I did some time ago. So I'm a research assistant at Noguchi Institute for Medical Research. And today I'll be presenting on the neglected dwarf tapeworm and its impact on deprived communities in Ghana. So I'm just going to give a short briefing about myself. So as I mentioned earlier, I'm a research assistant on an NTD, a neglected tropical disease research called 9. And I've been a research assistant on that project since 2018. My research areas are helminth studies and the human gut microbiome. And I'm also a scientific illustrator. So I post science comics on Instagram to help people understand all of the fun science stuff we do in our labs and how to get them to be interested in the science we do and to understand the things that we do. So on Instagram, Science Arts on Instagram, so you can always follow and then enjoy some of the comics I post. I served my national service as a research assistant in the Department of Parasitology, and that's where I still am. And I have a bachelor's degree in animal biology and conservation science. So just a brief history of Noguchi so that we all know what Noguchi is and what it is and what we do at Noguchi. So the Noguchi Memorial Institute for Medical Research is the leading biomedical research facility in Ghana here. And that's where we do a lot of our COVID testing now. And we do a lot of other research. We do research into bacteria, into parasites where I am. We do research into drugs and how to make new drugs for treating all of these diseases going on around us. We have virology departments where we go into study viruses, among other things. So Noguchi was established in 1979, and it was built in honor of Dr. Hideo Noguchi. So Dr. Hideo Noguchi is a Japanese who came to Ghana to do some research on yellow fever, but he ended up dying due to yellow fever, sadly. And I'm in the Department of Parasitology, where we conduct research into parasitic diseases like malaria, elephantiasis, bilharzia, helminth infections, all of these worm infections, among other things that we do. Every parasite, you can find almost every study related to parasites going on there. And we are ISO certified and SLEPTA recognized. So we have different research groups in the parasitology department, and my research group falls under the neglected tropical diseases or NTD research group. So now moving on to the main reason for this presentation. So we have this worm infection or helminth infection called Hymenolepiasis. So Hymenolepiasis is a cestode infection caused by the dwarf tapeworm Hymenolopus nana, or H. nana for short. And we all know tapeworm. I'm sure most of us have heard about tapeworm, but this is a different type of tapeworm because it's shorter. Its length is shorter than the actual tapeworm that we know. And it's called H. nana or Hymenolopus nana. It is found mostly in the tropics, and you won't really find it in cold climates. So you find it more in the tropics or subtropics. And there's a picture showing the adult Hymenolopus nana worm. And then there's the egg of H. nana, the Hymenolepiasis nana, as you see it under the microscope. And this infection is one infection which occurs with, you find it more when you are maybe looking at trying to focus on other worms, and then you find it in the stool samples. Sometimes you don't really find H. nana in single infection. You don't find people just infected with H. nana. Sometimes you find people, most of the time, you find people infected with H. nana and other helminths as well. Now, the life cycle of Hymenolopus nana, our dwarf tapeworm. So in the human, the host who gets infected, people cause all of this transmission of Hymenolopus nana or H. nana infection goes on when people defecate into the environment. So some studies have shown that Hymenolepiasis nana is a good indicator of fecal contamination in the environment. And once people defecate and somebody who is already infected with H. nana defecates in the environment, the eggs can go through different ways to infect another host who maybe might have let their guard down on sanitation rules. So when somebody defecates into the environment and maybe somebody ends up eating food that is contaminated with this feces or these eggs, they can also get the infection. People can also get the infection through an intermediate host. So someone can get the infection not directly, maybe by eating contaminated food, but can get the infection by ingesting an insect which has ingested the egg. So insects like beetles and fleas, when they pick up these, maybe they ingest the feces which contain these H. nana eggs, these eggs develop into a larval form called cysticercoid. So this cysticercoid develops in the insects, and then when an unsuspecting human goes in to maybe to take a cereal which has these insects in there, they end up getting infected by this cysticercoid, and then it moves on to the adult worm and then ends up producing eggs. But one fascinating thing about the Hymenolepis nana and its life cycle is that it has this part of its life cycle called autoinfection. So with the autoinfection, this bit of the cycle, the eggs don't necessarily have to come out. It takes the adult worm about 4 to 6 weeks to live in the human host. And then once with treatment and stuff, like even without treatment, within 6 weeks, it should be out of it should be dead. It should be dead. But one thing that keeps it going is that when some of these eggs, which are supposed to be ejected from your system through our stool of, let me say, feces, when these eggs remain in our intestine, they can still develop into the infective larval form. That's the cysticercoid. And this cysticercoid moves on to develop into an adult worm. And then they are hermaphrodites. So they don't need a male and female, right? They can reproduce on their own. So then they produce these eggs, and then they shed these eggs into the intestine again. And then when this infected person maybe defecates into the environment, they release some of these eggs. But the eggs that are not released can still go through the cycle again. So it causes this infection to persist over the years. So it makes treatment also very difficult, because once you don't treat people properly, you end up having some of these eggs present in their intestine, and then you keep having the adult worm developing, going through all the cycle over and over again. So when the adult worm dies, you still get another adult worm coming on board, because the eggs are still present in the system of humans. That's one thing that makes this infection very dicey, and the reason why I think we should give it more attention. Because of this part of autoinfection, we really have to give it more attention. So Hymenolepis nana is one of the most common intestinal helminth, which infects humans, especially children. So far, since 2016, when I started doing some field work and doing all of this stool analysis and then looking out for helminth, I've encountered Hymenolepis nana every time that I've been on the field since 2016. Yeah, 2016, I have encountered Hymenolepis nana. It is one of the most common ones that you see. So we have other worms like the round worms, the whipworms. You would see them, sometimes you see just a few of them, but for H. nana, you see them a lot. They occur frequently. And they could cause weakness, gastrointestinal discomforts, they cause itchy anus, they can also lead to poor appetite. It can also lead to poor appetite. And then one thing also is that they can also cause cognitive disability in children over the years. So that's one of the things about H. nana, and why we should also give it much attention, because it's causing the similar body just like other helminths. And H. nana has received less attention because it is usually in co-occurrence with other parasitic helminths. So just like I mentioned earlier, when you go into the field, you hardly see H. nana occurring alone. You see it occurring with diseases, with other helminths, which are of public health importance. And some of these helminths are like hookworm. So hookworm, Ascaris lumbricoides, or roundworms. These ones are the part of the NTDs, the neglected tropical diseases, the soil transmitted helminths. So people want to give them more attention. People want to give more funding to it. People want to think about how to treat these infections, but they don't really think that H. nana, they feel, okay, if I treat the others, it's a cover up for H. nana. They don't really give it so much attention. And for H. nana, there are specific drugs that can effectively work against it. But we have these mass drug administration programs which go on here in Ghana and some other parts of the world, where they give dewormers to children and in communities, even adults. They give them dewormers on a regular basis. So they have a time frame where they give them these dewormers. And it's all with the goal of eliminating some of these helminth infections from the system. But all of the drugs that they give, for, let's say, for hookworm, the worm that causes anemia in people who get infected by it, this worm is treated with a single dose of albendazole. That's what's the required dose from WHO. It's a single dose of albendazole. So when you give this albendazole to people in the community, like you just give it to them, because a mass administration, you just give it to people, you give it to them, they take their drugs. They work against these helminths, but then because the single dose of albendazole does not really work against the dwarf tapeworm, it becomes a problem. So you are treating the other worms, but then H. nana is still not being cleared from the system because that's not the required dose for it. And in a place where there is maybe schistosomiasis, that's bilharzia, they give people praziquantel. A lot of studies have shown that praziquantel is very effective against tapeworms, including H. nana. And if we are unlucky to have H. nana in a place where there's schistosomiasis, that means we can't clear H. nana. We are just clearing schistosomiasis, but because H. nana is not being it's not found in that area or that community, we can't clear it from the system. So then it makes it very dicey. So that means we have to rather give it a special attention and then focus on it and give drugs to people who have these infections. So in areas where H. nana is endemic, and it's everywhere. So it's everywhere. So it means while we are giving attention to other helminths, we should start focusing on H. nana and then maybe changing the dosage that we give to people so we can clear it out. And this is a distribution map of H. nana in the world. This one was taken in 2016. So the parts where there's the green, you see that it's all over, all from South America, all through Africa, and then to some parts of Asia. We have H. nana being endemic in these areas. And it's everywhere, like I said, it's everywhere. So we have to give it much attention. We can't just brush over it, saying that it's not really a neglected tropical disease. It's like, I think it's not a neglected tropical disease, so let me just let it go. We have to give it attention. So the objectives of this study the main goals were to determine the prevalence of H. nana infections, to investigate the impact of a single dose treatment with albendazole, because that is the drug, we give single dose for the mass drug administration program. So we wanted to see if this single dose would work against H. nana. Then if it works, that means we don't make a case against the treatment, we just keep quiet about it. So we wanted to investigate the impact of a single dose treatment with albendazole. Then we also wanted to find out the associated risk factors of infection and treatment. So this is our study area. So we worked in 3 communities. So it was a cross-sectional study. We just went in once and we took our samples, we treated people and stuff, all of those things. So this is a study area. So we worked around this area. Sorry, the arrow is not showing here. Sorry. So we worked around this area. So the community names, they're quite tough. So maybe as I go on, maybe I'll give a short form for it so that we all get it, so that we don't get lost along the way. So the 3 communities where we did a study was Jagbingbindo, Takumdo, and Wiai Chabo. I'll take it again. Jagbingbindo, Takumdo, and Wiai Chabo. They're big names, so don't worry. So maybe as I go along, I might say Jag, Tak, and Wiai. So those were the 3 communities where we did our study. So they were all around this area. So we sampled 566 people in the communities, the 3 communities together. And then we found out those who were positive for H. nana and those who were negative for H. nana. For those who were positive for H. nana, we treated them with a single dose of albendazole. And in 14 days, just like we did so one thing about this study was that I forgot to mention was that the bigger study was on hookworm. We were targeting hookworm, the worm that causes anemia. Yeah, so we were focusing on hookworm. But then I decided to look at H. nana and see what the story we could also tell concerning H. nana. So we treated those who were positive for H. nana, but for hookworm infection, we normally go in after 14 days of treatment to see how effective the drug has been. So we went in after 14 days to check those who were positive for H nana and those who turned out negative after treatment. And there's a picture of myself given administering albendazole in JAG or JAG-Bingbindle. Yes. Okay. So for the methodology, we use the Katokas stool preparation technique. So the Katokas stool preparation is a technique which helps us to prepare smears, stool smears on a microscope slide. And then we cover that stool smear with a cellophane. Yeah, we cover it with cellophane. So this cellophane has been soaked in malachite green so that it will clear all of the dangerous pathogens that could be present, and also helps you to view. It helps us to view the eggs if they are present. It helps us to view easily. So after preparing the stool smear on a microscope slide, we went on to use the light microscope for the diagnosing of these people. So we wanted to see if they had a helminth present or not. That's why we used light microscopy. So light microscopy helped us to see whether these people had the eggs present or not. So this is a picture of the egg of H. nana. There's a picture of the egg of H. nana as seen under the microscope. So this is what we saw. So once we see this, we note that the person is positive for H. nana. If we don't see it in the slide, we declare that the person is negative. And this reading was done by 3 different microscopists. So we have 2 trained microscopists who read different slides, and then we have a 3rd reader who does a quality check to make sure that they actually didn't see anything in there before we can be certain that this person is negative for H. nana or this person is positive for H. nana. Now, after doing all of that, preparing the stool smears and obtaining our data, we use some data analysis, equations formulas, and math formulas to calculate prevalence and cure rate. And then we use starter, the 14.0 version, for further analysis to find significance, and that's what helped us to determine the associated risk factors associated with H. nana. So prevalence was calculated by dividing the number of positive cases of H. nana by the studied population, that was 566, and multiplying by 100%. Then for the cure rate, to determine the cure rate of albendazole, we divided the number of people who passed treatment by the number of people over the number of people who were positive in the beginning, and multiply that by 100%. Now to the interesting bits, to the results, and I'll be explaining the results as I go on, so you would understand it better. So after calculating our prevalence, we wanted to look at the prevalence of helminth infection in the communities. So we didn't just go in looking for just H. nana. So as the study was a big study on hookworm, but we reported for other worms that we found. So hookworm was the most prevalent in the community. That had a prevalence of 19.61%. And hookworm is a neglected tropical disease. So in a community where we are finding this kind of prevalence, they were so interested in going in to do more research on hookworm, and they'll probably forget about H. nana because we are focusing on the neglected tropical disease and just leaving that one out, thinking, oh, I'll treat hookworm and then to get the other ones to be taken care of. So hookworm had a prevalence of 19.61%, followed by H. nana with a prevalence of 8.13%. And then this was followed by the whipworm T. tricura, which had a prevalence of 1.24%. And then we had Ascaris lumbricoides, that's the roundworm, having a prevalence of 0.71%. And then Tinea species, that's the tapeworm, having a prevalence of 0.18% in the community. We tried to find out if there was any significant association between these helminths, but we found that nothing. The p-value was greater than 0.05, so there was no association between these helminths. Now we went further to focus on the prevalence of H. nana in the 3 communities. And for a community like JAG-Bingbindle or JAG, we had the highest prevalence in that area, a prevalence of over 10% in Jagbingbindo. And then for Takumdo, it followed with the next highest prevalence. And then we move on to, no, sorry, we move from Jagbingbindo to Riai Chabo, which are the next highest prevalence of, of 6.5%. And then Takumdo had a prevalence of 5.8%. And I'll be showing the spatial distribution of these infections in the communities and explain the probability, what could be the reason why we found this level of prevalence in the different communities. So we actually got the GIS coordinates for every household that we visited. So this was we did a random sampling in different households. So for every household, we had a number of participants we got from there. And we got all their GIS coordinates and then we plotted them on the Google map, and then we got very nice plots. So we got some spatial distributions of H. nana. So in Jagbingbindo, this is the spatial distribution of Hymenolepiasis nana in the community. So the different points represent households, and the red one represents those that were positive, and the green represents the negative households. Now you see from here, and this is the actual picture on the ground, how the points look so clumped together. Now, when you go into the Jagbingbindo community, the houses are very close to each other. And studies have shown that in places where settlements are so close to each other, the probability of high transmission the probability of transmission of H. nana is very high in these places. So in places where you have houses close together, they are able to easily contaminate each other. So that could explain why Jagbingbindo had the highest prevalence in this study. That's why they had that highest prevalence in this study. And then we move on to Riai Chabo. In Riai Chabo, which had the next highest prevalence, for that area, we don't really find houses close together, and it's just as it is on the ground. Houses were not so packed to each other like it was seen in Jagbingbindo. And Takumdo, we had this kind of spread too, and we had a few households being positive. So probably because the households are few, that's why we are not really, maybe at the time of the study, we hadn't really seen transmission, but we don't know about the story on the ground at the moment. So one thing about these communities was that from the data we collected, the demographic data we collected from them, we realized that most of them practiced open defecation. And open defecation is when they just go into the place or just find a place in the community and then just defecate. So they didn't, just a few people were using latrines, pit latrines in the community, but the majority were just defecating into the environment. And studies have shown that in places where there is fecal contamination in the environment, there is a high prevalence of H. nana in these communities. And that could explain why, even in the places where we have this spacious households, we still find H. nana present. Because the percentage of people that were using open fields, open defecation, just going into the bushes or just defecating around, the percentage was 90.6%. 90.6% of the people out of the 566 people that we sampled, 513 of them were practicing open defecation. And we had just 46 out of the 566 people using pit latrines. That's like a proper form of washroom. Let me put it that way, a proper form of washroom. So that is quite alarming. That's quite alarming. That explains why we had this prevalence and the occurrence of H. nana in the communities. Now we went on to look further at age and gender to understand how H. nana was distributed within the different age groups and then the different sexes. So in males who were less than 16 years, they recorded the highest prevalence of H. nana, and followed by females who were less than 16 years. So the males who were less than 16 years recorded a prevalence of 52.17%, followed by females recording a prevalence of 32.61%. And then when we come to those who were above 16 years, we have females recording the highest prevalence of 10.87%, followed by males recording a prevalence of 4.35%. Now, children, this picture is actually what has been observed in so many studies. Children who are less than 16 years are more likely to play in the sand. There are children who will be playing more in the sand, and they would end up ingesting some of the sand along the way. They are the group that would hardly adhere to sanitation rules, and that explains why the prevalence of H. nana is high within this group. And you see that those who are who were more than 16 years of age, they are recording a lower prevalence of H. nana, because within this age group, you don't expect a lot of children to be playing in the sand. You don't expect a lot of eating of sand and people not adhering to sanitation rules. You expect this group to understand washing of hands and being hygienic. But within this group, you don't see that. And that's the reason why we had this high prevalence of H. nana. So for this study, we took some anthropometric data. We recorded the height and the weight of people, and we used that for calculating the body mass indices. So for the body mass index, it tells if a person is malnourished or not, depending on the value of their BMI. So for people who have a BMI a body mass index of less than 18.5, these people are categorized as being underweight. And people who have a BMI of 18.5 to 24.9 kilogram per meter square, they are classified as being normal, having normal weight. And those who have a BMI of 25.0 within 25.0 to 29.9 kilogram per meter square, they are regarded as being overweight. And those who have a BMI higher than 30 kilogram per meter squared are regarded as being obese. So we recorded this for the people that we did our sampling from, and we realized that a higher percentage of people were underweight. A higher percentage of people were underweight. Over 60% of the population we sampled were underweight, followed by a good number being normal. And then we had just a few people being overweight and being obese. But the one thing that we found was that out of those who were underweight, out of the group that was underweight, 91.3% of this group were positive for H. nana. 91.3% were positive for H. nana. And we went on further to look at some other dynamics within this group and realized that children who were less than 16 years were also very common. They were the most abundant people within this group. Children who were less than 16 years, they were the most abundant people within this group. And 91.3% of that group were H. nana positive. Now, from some studies that we found, and one study that we found realized that there is an explanation as to why those who are underweight would have an abundance of helminth infections within them, and I'll explain that as we go on. But for now, the one thing, the take home is that 91.3%, a higher percentage of the underweight group were positive for H. nana. So we wanted to find out which factors or what was causing the likelihood of somebody developing or getting infected by H. nana. And we found out that the significant variables were children who were less than 16 years and children who were underweight. So for children who were less than 16 years, they were the ones who had the greatest likelihood of being infected with H. nana. They were significant at a p-value less than 0.05, these different variables. And some studies have shown that children who are less than 16 years, they may produce some pro-inflammatory mediators. So before any inflammation occurs, our body has a way of producing these some kind of cytokines, some kind of, let me put it so we all understand, some kind of like chemicals in our system. And these chemicals or indicators have a way, they include the interleukin 1, the interleukin 6, and some other ones. And these interleukins have a way of impairing the way the body breaks down nutrients. So children within this age group are being affected by the fact that their body is producing some sort of chemicals or cytokines, simply that with cytokines, which can impair how their body breaks down nutrients. And this is the reason why even when helminth infection is not so severe within them, it can cause them to be underweight because their body is unable to break down nutrients already. And now they have these helminth infections coming on board. So then it causes them to end up being underweight. So that could explain that. But children within this who are less than 16 years play more in the sand. In the communities, when you go into the community, you see these children playing a lot in the sand. Their hands are dirty, but it's still going into their mouth, and they are eating even without washing their hands. So that explains, it's very understandable that we find this variable being significantly associated with being H positive. And those who are underweight, as I explained earlier, they are also one of the variables that are significantly associated with being H positive. So in the end, initially we were expecting to find maybe the lack of latrines to have an impact on somebody's positivity for H, but we didn't find that. So, but these ones also match up with what has been reported in literature. Now, moving on to the treatment of hymenolepiasis. So, for one to eliminate H, we either use 600 milligram of praziquantel, and these are recommended doses by WHO. So it's either 600 milligram of praziquantel or 400, and then let me put it well so we all get it. So the right dose or the right drug for the treatment of hymenolepiasis is 600 milligram of praziquantel. It's 600 milligram of praziquantel. But some studies have also shown that giving albendazole, single dose of albendazole for 3 consecutive days can also help to clear H to a higher extent. So the actual drug is praziquantel, but once you give albendazole for 3 consecutive days, it can also help you to clear H, and it has been proven in several studies. However, praziquantel is a drug which is only administered for mass deworming when in a community where there is bilharzia or schistosomiasis. So in a community where there is no schistosomiasis, there is no mass administration of praziquantel. So it doesn't matter whether we have H in the system. Once there is no schistosomiasis, nobody is administering praziquantel on a mass program. Yes, nobody is doing that. But then that is one of the drugs that has been shown to be very effective against hymenolepiasis because it's also caused by a tapeworm. And our mass drug administration programs, even in communities where we administer albendazole, it's only given as a single dose once and we are done. It's not given for 3 consecutive days. So you administer a single dose of albendazole on a one-time point, and then you give it to people, and then you are done. It doesn't help to clear H. It might clear the other worms, but it might not clear H. So after treatment, I mentioned that we went back after 14 days to take the stool samples of the people to check how effectively these helminth H had been cleared from their system. And we realized that Jagminbindo had the highest cure rate of 44.4%, and then Takumdo followed with a cure rate of 34.5%. And then Ria Chabo, those people in Ria Chabo who were infected with H were not cured after treatment. They all came back positive after treatment. And it has been shown that the efficacy of albendazole ranges from 45% to 100%. In published studies, have shown that it ranges from 45% to 100%. But this cure rate that we are recording is lower than that. It's lower than what has been reported as being like the okay range. So it tells us that, well, if we want to give it much attention, we should. And we should look at the treatment carefully and know how we are going to treat people and get this cleared from their system. If you want to pay attention to it, maybe we'll have to think again about the kind of treatment we are giving them. Maybe think about whether to introduce praziquantel in places where there is no schistosomiasis, just so we can get H. nana out of the communities, or maybe give albendazole on 3 consecutive times so that we can get people like cured, so that we can actually fall within the desirable range for a good efficacy of albendazole. We can fall within that range. And this observed cure rate, 0% in VI Chapel. Well, in the district where we worked, where these communities are, is the Kwandai district. The Kwandai district has been administering ivermectin, which is for onchocerciasis. It has been administering ivermectin to people in the community over the years. And sometimes one thing about these drugs is that they have very common points of mutation in their hosts. So if you give these drugs over time and people develop resistance to the drugs, they can confer resistance onto other things. So you're given the drug, maybe you're given ivermectin in the community over the years, and people start developing resistance. The sites where these resistances are occurring in the host can be transferred to other helminths, such that once you administer albendazole to people, you don't get them treated because the body has developed resistance to ivermectin, and it's a common site. So it develops resistance to ivermectin, and then this resistance is conferred on other things too, like on albendazole as well. So you end up treating people with albendazole, and they come back not really cured. We went on further to look at what was the risk factor that was associated with treatment failure. Why some of these people were not treated successfully, why some people still came back positive. And we realized from our analysis, the significantly associated variable was being underweight. So now underweighted children come back again. So we realized that children who are underweight were the ones who were very likely to fail treatment. So we have this variable falling back into place again. And some studies have shown that people who are underweight, they eliminate drugs faster from their system as compared to people who are obese. So in the end, when you give them a drug, the probability of having some amount of the drug, a desirable amount of the drug in the system to clear the worm is very low. You don't have enough of the drug that you administered in their system. So then you'd treat all of these people, but underweight people will still come back being positive because you've given them albendazole or whatever drug, but their system has released or eliminated a high part of these drugs. So whatever component of it is left is unable to clear the worm effectively. So that could explain why the underweighted people came back positive after treatment. That could explain why they came back positive after treatment, because the minimum free concentration of the drug in their system after treatment was so low, so it couldn't clear our H. nana. So those were the findings we obtained for this study, but we had a limitation for this whole study. Because our focus was on hookworm, the initial project was focused on hookworm, we went into the community and then we were checking for intensities of hookworm. But for H. nana, we were just checking for So we went into the community in that we were doing for H. nana was checking for the presence or the absence of H. nana. And as a result, we can't really tell if the low cure rates were because there were high intensity of H. nana. So that means the air counts of people who were infected was very high. Because sometimes when the eggs of the parasite are a lot in the system or in the intestine of the person, it is very hard for the drug to clear everything. So it can clear some amount of it, but not everything. So probably, so now we can't answer this bit of the question, whether our low response to albendazole was because of high intensities. So that was one of the limitations of this study. But in future studies, we did look at the intensities of H. nana in the community. And this limitation that we had explains what I was saying earlier, that all of these mass drug administration programs are looking at these neglected tropical diseases. They're looking at a particular group of helminths that are of public health importance to them. So we don't even care about these ones. And that was how we also made that mistake of looking at focusing on hook. And that was our main focus anyway. But we left H. nana out by not counting the number of eggs that were present on each slide that we prepared. So that was one of the limitations of this study. But our study was still successful. We still have had some good information that could also inform policies, could inform strategies of treatment. So in conclusion, just to summarize everything I've said earlier, realize that the prevalence of H. nana varied between the three communities, and that was explained by the reason that they had their houses so close together. So there was easy transmission of Hymenolepiasis nana in the community. And also because these communities lacked pit latrines, the majority of them were just defecating in the community. And as several studies have shown, in places where there is a high occurrence of open defecation, there is also a high prevalence of H. nana. From the findings of this study, tells us that MDA, the mass drug administration for lower age groups, children below 16 years, should be given more attention. It should be given more attention because we realize that they are the ones recording more of these helminths in there. And these helminths have a negative impact on the growth of these children, both mentally and physically. So there's a need for us to pay close attention to them and find a way to treat them effectively for hymenolepiasis. Also, efforts should be made to reduce indiscriminate defecation in the communities to halt the transmission of the intestinal parasites. So we should try to enforce proper sanitation. We should enforce the use of pit latrines. That's one thing that should be done in these communities so that we can reduce the amount of level of transmission of H. nana going on in the community. And also, we can also map out the prevalence of H. nana and albendazole cure rate in Ghana. And this can help us to know areas which are endemic for H. nana and areas which are not, so that we can go in with praziquantel, the most effective drug. We can go in with this drug and then treat people within these areas. But once we don't really have any information on the prevalence of H. nana in the different communities, we can't really know how to administer treatment, because we know praziquantel is given to people, it's given in communities where they are endemic for schistosomiasis or let me say bilharzia, but it's not given in other places where there's just H. nana. So it's necessary for us to have a database where we can tell the prevalence of H. nana in different communities so that we can change our strategy, our treatment strategies, and go in there and treat these people with praziquantel as well, just like we do for schistosomiasis. I would like to acknowledge the leaders, people, and health management team of the three communities where we carried out this study. They were really helpful in helping us to talk to the people and to get them on board for this study. And the Wilson Lab team, of which I'm a member, I'm a research assistant on this team. and the Department of Parasitology from Noguchi, where I am based. And this study was in collaboration with Yale University. They were focused on the hookworm bit, yes, but they were all part of this collaboration. And the funding was from NIH. It was awarded to Professor Michael Wilson, and our one award was offered to him, and this is how come this study was possible. Thank you very much for lending me your ears and taking part in this interesting presentation. Thank you.
That was fantastic. I was I was really excited, and that lived up to it. That's fantastic. All right.
Thank you.
So first of all, again, wonderful talk. So what we'll do now is I'll just go ahead and jump straight into the questions. I had a number of them, and a few other people did as well. So some of these are. So I'm a chemical engineer, so I have a lot of ignorance in the medical area. So forgive me if these are very simple questions. But let's. So my first one was: would you say is it a realistic goal to try and actually wipe out something like H. nana, either whether it's on a country scale, a continent scale, or even ideally a worldwide scale?
Yes, it is possible.
Okay.
It's actually possible, because it's hard, even for these neglected tropical diseases, including hookworm infection and schistosomiasis or bilharzia. We were hoping, well, the world was hoping to eliminate these infections in 2020, by 2020. But now they've set out a new roadmap to eliminate it by 2030.
Okay.
Because of some of the hindrances, some of the problems that go into all of these strategies, these controlling measures and other things. But it is still possible. That's why I mentioned the mapping out. So once we go into different communities for this kind of map, where we go in there, we take 2 samples from people, we go through the diagnosing to find out if they are positive for H. nana or not. We can tell the prevalence of H. nana in the communities. And as a result, we can go in there with the right treatment to then get it out. And then also, when we have this information, we can then educate our health professionals in the communities to enforce the use of pit latrines instead of open defecation. But once we have no idea what is happening on the ground, we have no idea the kind of helminths that are present, we can't actually do anything about it. But it is very possible that we can get it out of these communities.
Okay. Interesting. I just had a question and it completely went away from me. All right, I'll move on to the next one I actually wrote down. So have you, in your experience, have you encountered resistance to any of the testing or the treatment? And if you have, how do you kind of work with the communities to try and improve compliance, whether it's with things like using pit latrines or even just taking the medicines that will help, get rid of an infection or prevent the sort of the cycles that you talked about, where once somebody has it, then it sort of spreads in the community and all that. How do you deal with something like that?
Okay, so for now, I can't. So far, for all the field studies we've done, we can't specifically say that people have developed resistance to the drug. But sometimes when you go into the community, people are resistant to taking the drug. Sometimes you have people who don't want to take the drug, even when you explain to them what the cause of the infection is. And mostly it's because some of these people are not educated, so they don't really see the impact of the worms. Because you tell somebody that you need to take this drug, it's very good, it will help to clear the worms from your system, and they're like, I'm fine, I can always go to the farm, I've been going to the farm, I'm always strong, I don't see why I should take a drug. You are telling me I'm sick, I'm telling you I'm strong. So we have this kind of problem with people. But sometimes the good thing with some of the communities is that they're really good, but the good thing is that sometimes when you go to the communities, for a recent study that we did, we went into the community and then we were also drawing blood and checking for measuring some parameters in the blood. And then we realized that some people were anemic. And some of them, it was like serious anemia. So we had to rush back to the community, get those people to the hospital, for the hospital to actually give them a stronger dose. Because for our study, we're not required to give more than a single dose of albendazole. So we took them to the hospitals. And after that, that's how the information started going around that Oh, so I almost got sick. I was really no, I was really sick. And these people actually helped me. Like the drug really worked. That's how I'm fine now. I almost died.
Okay.
So when they go in there with that information, now it gets people on board. So
Okay.
sometimes you meet people who are like, I don't want to take the drug. But sometimes when you get this kind of information, good information going on about the efficacy of the drug, it helps. It really helps. Yeah.
That's good. Okay. That makes a lot of sense too.
Yeah.
And I actually remembered the question that I wanted to ask.
Okay.
So you show the way if I understood correctly, the way that you actually see if somebody tests positive is you actually like take a fecal sample and look
Yes.
at it and see if you see the eggs. Is there any other way to test for something like H. nana that doesn't require, because it seems like that, if you were trying to check a large community or something like that, it seems very labor intensive, right? Because you have to prepare each slide and you have to actually go in and look for the egg and things like that. Is are there any other ways to do that check where you could,
Okay.
you
So
know what I
far,
mean?
yeah, I get you.
Mm.
So far, the gold standard for diagnosing people of helminth infections is microscopy,
Okay.
using a microscope. Yeah. So we prepare the stool slide. There are other ways of preparing the slide or getting your sample onto the slide and then viewing it in the microscope.
Hmm.
One other thing you can, I think once you keep doing it, it's not really so labor intensive, because in another paper that I read, they were
Hmm.
using colonoscopy for
Hmm.
checking for people who were positive for H. nana, but that one is even way labor
Oh
intensive.
yeah.
Yeah, that's way labor intensive because going into the gut of people, it's kind of
Right.
tough. Yeah, but I believe that if we set out to use biomarkers, and
Hmm.
currently my research that I'm doing is focusing on biomarkers using the gut microbiome or microRNAs as possible biomarkers of infections,
Hmm.
helminth infections. So I believe that if we're able to get specific biomarkers that can tell us if somebody is positive for H. nana or not, it can help us to
Okay.
be, and we don't go through all of this microscopy. Yes,
Right.
we
Right.
can
Right.
use just a part of the sample and then maybe place it on maybe an equipment or something, and then it quickly tells us that this person is positive for H. nana. But for now, the research hasn't really gotten to that level. We haven't really gotten any kind of equipment or any kind of
Hmm.
diagnosing tool that can tell us without going through all of this microscopy, preparation of stool slide and stuff like that. But
I got
I
you.
believe in the coming years there would be some research on that. Yeah.
Okay, that's it. Yeah, I mean, certainly it's not an easy thing to do. I was just
Yeah.
curious if it
Yeah.
was even feasible. So that's exciting to hear that there's at least a good shot of it.
Yeah.
Okay,
Definitely.
so you talked about the intercommunity spread.
Yeah.
Is water contamination an issue for the spread of H. nana? So like not necessarily like somebody defecates near like a stream, and then somebody else drinks out of the stream, but like one of the issues that we have with especially chemicals here in the US is there will be somebody will like bury it under the ground, and then it gets into the water table, and then it
Yeah.
shows up in somebody's water.
Yeah.
Is that an issue, or is it more the sort of immediate spread within the community?
Well, I have seen papers. We didn't look at we didn't check their water, but I have seen papers that have reported contamination
Hmm.
through water. Yes. So
Okay.
they've checked some of the maybe some their taps, the water running through their taps. They've gone for that kind of water, and they've checked and they found out some to be positive, have some H. nana X in there. But we didn't do that. We
Okay.
didn't do that. But that's one way, but it's not really it's you might get in H. nana through that, but it's not a very likely way that you can get H. nana. Yeah, but it is
Okay.
possible for there to be contamination in the water. But then once there is the egg present and it's still viable, and somebody takes in that water, they can still get infected by H. nana.
Okay. And how long is the egg viable? Like what's the timeline for that?
I get I let me just check quickly.
Sure.
Um, from what I read, The egg should be viable, I think, for about 2 weeks or so. I
About
can't
two
really
weeks.
yeah, I think about, I have to check again, but
Oh, that's
yeah,
fine. Yeah, just I was looking for just a general. I was just curious.
yeah,
Okay. Yeah, so I could see where that even yeah I could see where the in the water table maybe it's not as big of an issue because it might take a while, but
wow,
certainly still
before, yeah,
possible.
yes,
Okay. So in the data that you showed on the cure rates,
yep,
we Chabor had the 0%, and I think
yeah.
you kind of touched on why you thought that was, but could you elaborate on that a little bit more as to why their cure rate was so much worse than the other two communities?
Okay, so one thing I mentioned was that these communities already have ivermectin treatments going on there.
Hmm.
So one of these mass drug administration programs, they administer ivermectin for the treatment of onchocerciasis in these communities.
Okay.
So yeah, already they have this drug going on. And papers have shown that a paper that was published, also a study going which took place in these communities,
Hmm.
mentioned that VI Chabo was one of the communities that were poorly responding to ivermectin treatment.
Ah.
And that could be because I can't really say for sure, but that could be because they are developing resistance to ivermectin. I can't really say, but or maybe there is something going on in Chabo that is causing people to respond poorly to ivermectin.
Hmm.
And one thing about these anti-helminthics is that they have a common site where they cause resistance. So if the gene that would
Oh.
cause resistance for to cause resistance to ivermectin will be the same gene that will cause resistance to albendazole. So if I'm not responding to
Oh.
ivermectin, then I'm also not going to respond to albendazole. So
Interesting.
that could be the reason why VIH was giving us that zero cure rate. Yeah.
That's interesting. Okay, because that's it's almost like I mean I guess it's a similar problem to antibiotic resistance.
Yeah.
Okay, that's interesting. I mean it makes sense that it could happen. It's just not something that you ever really think about.
Yeah.
That's fascinating. Okay. So for you talked about how one of the reasons or one of the hypotheses for why the drug treatments aren't as effective on underweight people is that the medicine basically passes through so quickly that it's not fully
Yeah.
effective.
Yeah.
Are there are there ways to change how you actually administer the drug that might make it more effective, or
I think
is that a possibility?
I think there is a possibility. I think we would have to, for our study, we were just given single doses of these drugs.
Hmm.
Probably praziquantel would be more stronger at clearing it than albendazole.
Okay.
Or we could also try and focus on getting this kind of these malnourished children back
Hmm.
to being normal.
Yeah.
Then we offer them treatment. Yes. So because if the child is still underweight and you're still giving the drug, it's still going to be a waste of drugs. So probably the best thing will be to move these children from the state of being malnourished to being
Hmm.
normal. Then they can offer them treatment. So that could be the way to go about it. Yeah.
Okay. So is it sort of is it kind of part of a cycle where like maybe you start with a child that's at a normal body weight, they get they contract H nana, and then it makes them sick or not? Yeah, it makes them sick. They become more underweight because it's affecting the way the body breaks down the nutrients, and so then even if you clear it out,
Yeah.
are they now more susceptible to it? Is that kind of one of the things that you have to watch out for?
Yes, yes. So those who
Okay.
are underweight are more susceptible to the infection.
Okay.
So they are unable, children who are less than 16 years are develop these kind of chemical cytokines which affect how they break down nutrients.
Hmm.
So already there is that one going on, and there is a presence of this helminth infection. So once that it becomes a problem, it moves them towards the state of being underweight, and then you end up having these helminths being proliferating in the system
Yeah.
because they find that as a good ground for them because you are treating and they will never get out. So they find that as a good ground to proliferate. So
That's interesting.
yeah,
It's interesting to see how there, it's all these different problems that kind of come together, and you can't
yeah,
treat just one. You kind of have to handle multiple issues at the same
yeah. You
time.
have to yeah.
That's interesting.
And
Uh,
I believe that the way to go
mhm.
with treatment of any kind of disease, like you have to look at different angles. Otherwise, you might just take one angle and then end up coming back to the same issue again.
Right, right. Is there any preventative medication available to like could you take a child before they've ever been infected with H. nana? Is there anything that you can give them to make it, maybe it doesn't prevent an infection, but makes it less likely that they'll be infected, even if they ingest it?
Okay, so far there's nothing. I don't know of anything like that.
Hmm.
Yeah, I don't know of anything that you can give to them that
Okay.
would, yeah, reduce. Probably maybe if there's a study that would look at maybe developing vaccines against these helminths, probably that
Sure.
would help. Yeah, but so far we don't have that. So it's
Okay.
just maybe keeping children maybe from playing. And it's like all of these things have a negative and they have a positive aspect, because you're telling
Hmm.
the child not to play too much in the sand.
Right.
But if you look at the hygiene hypothesis, like it kind of helps, but too much of it becomes bad.
Sure.
Yeah, so there's that interplay.
Yeah, it's a difficult balance that you have to
Yeah.
strike.
Okay.
So we had a question here from Fat Gamer 97, and wanted to know. Yeah, with more drug administration, maybe like wider spread and possibly maybe better targeted, would it be possible to completely clear the helminth infection? Do you think?
Okay, I believe it's possible. Like there's that possibility.
Hmm.
Yes, and all of this is not really. It shouldn't be, the focus shouldn't be solely on the drugs. The drugs will do their bit, but once you don't take care of other environmental factors, you still have people picking up these infections again. So you
Right.
can clear a whole community by the administration of these drugs, but in the end, you still have people going back for this infection because their habits have not changed, the way
Right.
they do things have not changed. Yeah. So the drugs will work, but we also have to target other things. You have to look at other areas too, so that we can work holistically and then get it out of the communities.
Yeah, absolutely. It sounds based on what you've talked about. It sounds like the hygiene, especially, seems like a big thing. Like you said, pit latrines or
Yeah.
some kind of sewage system or something like that would
Yeah.
be a big first step.
Yeah.
But it's like you said, it's interesting. The multi-pronged approach that's going to be necessary
Yeah.
is it's an interesting but very difficult question. Um,
Yeah, it is.
yeah. So I think I'll give a few more, just a little bit more time for any last questions to come in. But I'll go ahead
Okay.
and preemptively say thank you again. Really interesting
Thank you
talk.
so.
Uh,
Thank you.
I personally learned a lot. 'Cause, like I said, not remotely an area that I'm familiar with, and so this was
Sure.
all of this, both the background and the actual field work that you did were fascinating. And so I really appreciate you taking the time. I know is it what, I guess it's probably almost 4:30, 4:45 there.
Yeah.
Oh man, then I won't keep you. I won't keep you too long. I really appreciate you taking the time. Then,
It's my pleasure.
but yeah. So I think that's all the questions that we've got. So I will say thank you one more time to Lydia.
Okay.
Like I said, fascinating work. She is on Twitter as well. I'll tag her in a tweet later tonight once I have the video uploaded to YouTube. So if you found this work interesting, make sure you give her a follow. As she also noted, she has the Scienty Art Twitter account as well. I'll make sure to tag that one as well.
Yeah.
So give her a follow if you found this work interesting. And thanks to everybody for coming out. Thank you again to Lydia for the fantastic talk. We are still looking for somebody for next week. So if you know somebody that wants to give a talk, please do let me know.
Sure. I will.
Thank you. And Otherwise, have a great weekend, everybody. Please stay safe, wear a mask. COVID will eventually go away if we actually do what we need to do. And I will see you all next week. Yeah, I'll drop the link for the Discord one more time in case anybody in chat wants to join it. And I think with that, I think that's it. So thank you again, everybody, for coming, and we'll see you next week.